Good afternoon, and welcome to the latest of the Kavli Foundation Spotlight Live webcast, where we hear from scientists on the cutting edge of discovery. Today I'll be speaking with three neuroscientists who are among the first in the world to study how the Zika virus damages the developing brain. Just a few months ago, the virus wasn't on the radar of most scientists, let alone brain researchers. Then, as we all know, officials in Brazil reported a surge in the number of babies born with unusually small heads, a condition known as microcephaly. That surge seemed to be related to an outbreak of the Zika virus. Was Zika the culprit? No one could say for sure, but my guests were quick to realize that they had the tools to help figure that out. They set about testing if and how the virus infects developing brain cells and what happens when it does. Their findings, published in rapid succession this spring, have helped establish the link between Zika and microcephaly, and they're learning more about the effects of the virus on the brain every day. Over the next 30 minutes, we'll talk about the virus's tactics. We'll also find out what it's been like to work on what the nation's top infectious disease doctor has called an unprecedented threat. My name is Lindsay Borthwick, and I cover neuroscience for the Kavli Foundation. With me today is Arnold Kriegstein, who is both a scientist and a neurologist at the University of California, San Francisco. Dr. Kriegstein studies how stem cells mature into neurons, including the signals that spur those cells to multiply and form the brain's outer layer. He is a member of the Kavli Institute for Fundamental Neuroscience at UCSF. On the opposite coast, I have Hongjun and Guo-li Ming from Johns Hopkins School of Medicine in Baltimore. Dr. Ming's research is focused on understanding how brain cells migrate and make connections as they develop. Dr. Song studies the role of stem cells in the nervous system and whether abnormal development of these cells can lead to disorders such as autism and schizophrenia. They are both associate members of the Kavli Neuroscience Discovery Institute at Hopkins. Welcome, Arnold, Hongjun, and Guo-li. I'm thrilled to have you here today.
So are we.
Yeah.
Yep.
Here.
A quick reminder to our audience before we get started. If you have questions for our panel, please send it by email to info@kavlifoundation.org or tweet us using the hashtag KavliLive. We'll get to as many questions as we can. Okay, to get started, I'm wondering, as neuroscientists, what questions immediately sprang to mind when you first heard about Zika and the possible link to microcephaly in newborns? Professor Song, would you mind starting?
So when we actually first heard the news, we saw the first reaction actually is maybe does not do much with the nervous system. It may be something affect the immune system and eventually affect the fetal development. So the first question come to my mind, actually many people in our labs, is whether the virus can directly deal with the nervous system or not. And really, it's the data from the clinical study to show that the virus actually can enter the fetus, get us started, to really ask the question whether the virus can directly infect the cells in the nervous system, what exactly cell type they can infect.
Okay. Anybody want to add to that?
So my approach was a little different. I have a lab that's really focused on studying human cortical development, and so we've also been very interested in microcephaly. And when I heard that the Zika virus was linked to the possibility of human microcephaly, we became really quite interested and started going through the literature. And I have to say, luckily, there wasn't very much literature on Zika at all. But one of the papers described how the Zika virus enters skin cells, and it highlighted the role of a particular entry factor or receptor called the AXL receptor. And this rang a bell because we've been doing single-cell gene profiling of developing cells in the human brain, and this particular protein, the AXL receptor, was very, very highly expressed in the neural stem cells, which are exactly the cells you'd expect to be targeted in the production of a microcephalic patient, somebody who has a small brain. And so we started looking more carefully at the expression pattern of that particular receptor and became more and more impressed at the role it probably plays in producing not only the microcephaly, but some of the other clinical symptoms that people have described in these babies that were born in Brazil with Zika-induced microcephaly. So that's where our interest really started.
Okay.
So, yes.
Go ahead.
So for me, it's also very similar. It's the microcephaly actually caught my eye, as we are trying to develop this 3D mini brain cultures in dish, and we thought that could be a perfect system to study how it might cause microcephaly. So that's our initiation.
Okay, great. I mean, all three of you have touched on things that I want to bring up in the course of this discussion, including the mini brains, the cerebral organoids that you just mentioned, Dr. Ming, and also the AXL protein, Dr. Kriegstein. My next question, though, is for Professor Ming. In a paper you published in late March, you showed that the virus can infect human embryonic cortical neural progenitor cells. That's a bit of a mouthful. Can you tell me more about what these progenitor cells are and what they do in the brain?
So I actually, to my mind, Dr. Kriegstein probably is the best person to answer what is this neuroprogenitor cells.
Okay.
He's the best person to describe this.
Okay, I'll pass the baton to you then.
Well, so I appreciate that. Thank you, Golin. So it turns out that the neural stem cells in the developing brain are cells that are known as radioglia. And they have several different functions, but one of them is, in fact, to produce the neural cells, including neurons and also the glial cells in the developing brain. And they derive from neuroepithelial cells, which are the earlier version of these radioglial cells. And so that lineage, the neuroepithelial cells and then the radioglial cells, are the neural stem cells of the developing nervous system. And in the cortex, they produce, of course, all the cortical layers. And for microcephalic patients, and including the patients who suffer from Zika and microcephaly, it's the cortex that seems to bear the burden of the insult. And so we and others, all of us here, were really interested in those neural stem cells, the radioglial cells, and how they could be targeted in this disease.
Right, so these radioglial cells mature into neurons eventually and migrate out to form the outer layer of the brain. Is that correct?
Well, not quite. They don't mature into neurons. They actually produce daughter cells.
All right.
And those daughters are actually a different set of progenitors. They're referred to, we call them sometimes intermediate progenitors, or sometimes they're basal progenitors, but they're a different cell type that is still a progenitor, still cycling, dividing cell. And they seem to produce most of the neurons, especially in the human brain. And so there's a two-step process where the radioglial cells are the neural stem cells. They make a more limited type of progenitor that then makes the actual neurons. And you're right, they do migrate along the radioglial fibers to get to the brain.
Okay. So Professor Ming, you grow these cells in the laboratory, and when you look at them under a microscope, what happened to them as a result of Zika infection?
So the first reason we actually focus on neural stem cells, because in genetic forms of microcephaly, these are the hypothesized cells to be affected the most, that results in genetic forms of microcephaly. So that's why we also try to focus on these neural stem cells or neuroprogenitor cells in cultures. They are not like as strict as the developing brain they're described. But anyways, so when we put the Zika virus, actually very low dose of Zika virus, less than 10% of the virus in the dish, and we look at the cells after like 56 hours or 2 days or so, and we found surprisingly it's not just the 10% of the cells, neuroprogenitors, are infected by the Zika virus. Actually, we found it's like 80% of the cells are infected by the Zika virus. That suggested to us this is actually a productive infection, meaning that the infected cells become a viral factory and they produce more viral particles and being released to the media, and that will continue to infect other neuroprogenitor cells in dish. So that's kind of scary for us.
Right, it's like a cycle that just keeps continuing.
Yes.
Okay, Professor Song, in another set of experiments, you looked at how Zika affects clusters of developing brain cells that are called cerebral organoids or mini brains. Can you describe these organoids for our audience? I mean, how closely do they resemble a developing human brain?
So this is a new technology happens in the last few years, where for many years people have been differentiated cells of the stem cells, including human stem cells, in 2D cultures or monolayer cultures. Of course, the caveat of that study is they are not as organized as in the in vivo situation.
Okay.
So over years now, people actually develop a 3D culture, and that allow you to have some kind of structure mimicking what happened in vivo. So this has been done for many different tissues, kidney and liver, or others. And really, it was the breakthrough of multiple pioneers in this field, and they actually showed you can also do this for the brain. And so basically now, people have methodologies allow you to generate a 3D structure, has a different layers structure in a way where you have stem cells, radioglial cells, you have immature, intermediate precursor cells, and immature neurons. So basically, this is a growing 3D structure mimicking the fetus in some degree in the dish where we can study. You point a very good question, which is how similar of that to the real fetal brain. I think that's really is an ongoing task. People are really keep trying to make them better. There's different protocols out there, and I think each one of them has its strengths and weakness. And we started by improving the technology. So that's actually what I've been working on at Guo-li's lab and my lab I've been working on for the past 3 years.
Okay, but so what you have is a cluster of cells, different types of brain cells, and they are organized to some extent in the way that they might be in a developing brain. Okay, so then.
Well, actually, so may I add on top of that? Actually, at the early stages, they are amazingly similar what happens people see in early embryos.
Okay.
So the stem cell layers and the immature layers, they actually form really, really well. I think is the difficulty is when they grow longer over time, we lose some of control.
Right. So early on, they're a fairly good proxy. Okay. And so you infected these mini brains with the Zika virus. What did you find?
So actually, one of the motivation why we want to do this is, of course, in 2D cultures, you can look at which cell type a target of Zika virus and what happens. But really, we can't really look at microcephaly per se, because microcephaly is the center of the cortical layers. So that we need a model to study, and that's the reason we did with the Zika virus in organoid. And what we found is quite interesting. We actually did a couple set of experiments, and the first thing we saw is because we grow the organoid, we can model the exposure of virus at different stage of pregnancy. So if we give virus early on to the organoid development, we would be modeling maybe the first trimester. And if we give the virus later on, we can mimic a later stage of pregnancy. And the effect actually really depends on when that happens. So if you apply the virus very early on, so the mimicking the first trimester, and what we find is all the cells are neural stem cells, when they get infected, and they basically stop the cell from sort of slow them down to division, but also kill many of them. And this really leads to destruction of the whole organoid. And this pretty much mimicking maybe in humans as well, that probably is going to lead to the death of the fetus very early on. If we expose that at a later stage, and what we find is now, because the tissue structure is much bigger, we get a very low infection early on, very few cells are infected at early stage, but gradually, just like in dish, there are more and more cells becoming infected, because again, they turn them into a factory. And then gradually, this leads to a reduction of the stem cell division, and also death of not only the stem cells, but also neurons. We don't understand it yet, so clearly this is an indirect effect. But that really leads to an effect looks like center of the neural layer, just like the microcephaly, the center of cortical layer.
The cortical layers get thinner, okay, as a result. Okay.
Yep.
Dr. Kriegstein, so you work as a pediatric neurologist some of the time, and you've seen babies born with microcephaly due to genetic causes. What do the cases of microcephaly you see at the hospitals in San Francisco look like compared to the cases of Zika microcephaly that you've read about in the literature?
Well, I think you're actually touching on a very important point that's often missed. So the genetic forms of microcephaly, which I think it's fair to say most neurologists are more familiar with, they're very different than what we're seeing with this Zika virus microcephaly. So the congenital genetic forms actually produce a brain that structurally looks rather similar to a normal brain, except about a third of the normal size. So if you look at the external features, it's a folded brain. It has pretty much the same kind of landmarks that you'd see in a normal, fully developed sized brain, but it's much smaller. As I say, approximately a third of the normal size. That's the outcome in most of the genetic forms of microcephaly, and that's because the genetic defects affect the way these cells divide at early stages, the neural progenitors that we're talking about. And so you reduce the population of progenitors and you wind up with smaller numbers of neurons, fewer neurons, and then a smaller brain. But structurally it looks quite similar to a regular full-size brain. The problem with the Zika virus microcephaly is that it does several different things. One is it does target these progenitors, and so there are fewer cells making neurons than normal, and that would produce a smaller brain. But in addition, the virus destroys cells after they're created and produces a kind of necrosis, a death of the cells, not just in the progenitor region where the cells are dividing, but in the cortical plate or the mantle where the cells already have started to mature. And so there's degeneration in addition to just not developing normally. And this is a much more devastating disease. And if you look at the scans that are being published now from patients in Brazil, the cortical calcifications, the thinning of the cortex, the large ventricles, changes in the cerebellum and brainstem and other areas besides just the cortex. These are striking features that you don't see in the typical genetic forms of microcephaly. And I'm afraid that the outcome for these babies is going to be much worse.
Okay.
Yeah, actually, maybe I can add on top of
Sure.
that. That's actually something exactly as Arnold said. Is what we find is that in these organoid studies, we find the cells are also has sign of cell death. For example, they express the marker for cell death, but they are not positive for virus. So that's suggesting is the environment is really terribly destroyed by the virus. Now, even those cells are not infected, they are going to die. So this is much worse than sort of so-called cell autonomously, you have a genetic defect, only those cells will be affected. But this is not only affect the stem cells, but also their neighborhood.
Wow, okay. And Dr. Kriegstein, you've pinpointed a molecule that you mentioned earlier called AXL, which seems to allow the virus to enter the cells. Does it provide clues as to why the Zika microcephaly is so severe?
Well, yes and no. So I should mention that one of our real areas of interest is the mechanism for Zika-induced microcephaly, because if we understood that better, there may be strategies to protect these babies. So we're very interested in which cells are vulnerable. And the ones that express AXL fit into a very intriguing pattern. It's the radioglial cells that we've mentioned already, which are the neural stem cells. It's also the astrocytes, which are the lineage-related descendants. The radioglial cells turn into astrocytes, so it makes sense that they'd also be involved. It's also the microglial cells and the blood vessels, the endothelial cells, which form the microcapillaries of the developing brain. And this immediately raises a couple of issues about how the virus could be entering the brain, either through the radioglial cells, through the CSF, the spinal fluid that's in the ventricle that is actually in contact with these radioglial cells, or by a hematogenous route through the bloodstream, through the endothelial cells, which could also take up the virus very readily. And what was also interesting are the cells that don't express AXL, which include the neurons. So we know the neurons are dying in this disorder, but our hypothesis would be that they're not primarily attacked because they don't seem to express the necessary and sufficient receptor, the AXL receptor. And not only that, but the other types of progenitors, the intermediate progenitors I mentioned earlier, they also don't express the AXL receptor. So it looks very selective, and we would predict, based on this idea at least, that there are certain cells in the developing brain that are vulnerable and others that are not. And so we're very interested in testing that idea to see if it's true. The other difference is over time, there's a change in those cell types and in the expression of this receptor, so that a virus infecting a first trimester fetus is going to have a different outcome than one, obviously, that affects a fetus at later stages.
Mm-hmm.
These are all features that we're very interested in looking at.
So you mentioned you're interested in understanding the mechanism with the goal of potentially finding a treatment or a way to prevent these severe birth defects. Now, Anthony Fauci from the U.S. National Institutes of Health says it could take years to develop a vaccine against the virus. So what other strategies are you thinking about? And I'll ask all three of you because I know you each have some ideas on this, but perhaps Dr. Kriegstein can start.
Well, so since we published our first study, we now have access to the actual virus, and so we're very interested in doing some sort of a screen that we can use with an assay to look at infectivity to screen for compounds that might actually be able to protect the cells from actually being infected, or even after infection, prevent them from dying. And we're especially intrigued, as I mentioned, by the mechanism of entry of the virus. Because if we can block the entry of the virus to the cells of that are being destroyed, we could actually protect the babies. And we're working at UCSF in collaboration also with Susan Fisher, who's an expert on the placenta, because we think the placental barrier, that is to get from the mother to the fetus, you have to go through the placenta, might be another strategy for trying to protect the baby. And there may have commonalities between what's causing the virus to infect the placenta and what later on allows the virus to infect the fetal brain.
Guo-li or Hongjun?
Yes, I think obviously this is try to find ways to prevent Zika caused cell death or prevent that bad effects on our cells. It's a kind of obvious question, so we are actually doing some similar experiments, try to screen for effective molecules that can prevent the cell death or prevent the viral entry. So that's something we're doing currently right now.
Okay. I mean, what are the challenges, though, to treating the brain so early in development?
So one of our strategy has been, instead of testing a new drug, which is going to take years in the clinic to develop, so our strategy has been just so-called repurposing drugs. So what I've been doing is doing the screen using the drugs already approved by FDA. So those drugs already have been tested in humans to be safe and used for other conditions. So we're now testing those drugs to see whether any of them have any antiviral effect or actually protective. Of course, there are also issues with that because most of drugs are approved for normal people, not for pregnancy for babies. Right. But at least they're one step closer.
Okay. So they've already sort of passed that first sort of safety test.
For normal people, yeah.
Right. Okay. Now, other than seeking a treatment and screening drugs and small compounds, what are the urgent questions you're still striving to answer about Zika and the developing brain?
Well, we're very interested in how the virus actually gains entry to the fetus in the first place, because there are several different possible routes of entry. One of them is through the amniotic fluid itself. Once the virus goes across the placenta, it could infect the amniotic fluid at very, very early stages, before the neural tube closes. That would allow direct access of the virus to these neural stem cells, the neuroepithelial cells we mentioned earlier.
Right.
But at later stages, once the fetus is more developed, it's possible that a hematogenous route, that is, if the virus got into the bloodstream, it could then spread to the brain through the capillary system. And I should mention that the phenotype, that is, the way that the virus affects the brain, might have a lot to do with the route of entry. And these mini brains, the organoids, which we are also using in our lab, they're absolutely fantastic. And the idea that you can take a skin cell and convert it into a little human brain is unbelievable. But they do have some limitations. And among those limitations are, first, they don't express all the cell types that you find in the normal developing human brain. And that includes the vascular system. So they don't have endothelial cells. And these microglial cells, which may be very important, aren't found there either. So in some way, you need to look at the whole organism, or something a little bit more resembling an actual intact human. And so there may be other animal models that would actually solve that problem. And so using non-human primates as a model, we're actually also exploring whether ferrets might be a good model. But and mice have been published just recently as a model for Zika.
Yeah,
so
I saw that.
Any of these in vivo models might help answer some of the questions that the organoids or the limited access to primary fetal tissue isn't going to be able to address directly.
Mm-hmm. Professor Ming, are there questions on your mind?
So for us, we're really trying to understand which part or which protein that encoded by this virus is causing the bad effects to the cells, to neuroprogenitor cells. So that's something we are currently working on, really trying to understand. Is that the envelope protein, is a different proteins produced by, or even RNAs produced as the Zika virus is an RNA virus, or even RNA are actually doing having some toxic effect to the cells. So we're really trying to understand the molecular mechanisms.
Okay. I guess the Zika virus is part of a family of viruses. Another member of that family is dengue, and it's been studied, I think, in more detail. Are there any clues from that kind of literature as to what might be the protein, as you mentioned, or the RNA or whatever component of the virus that's triggering this?
So we actually have been doing direct comparison in the same system to look at the impact of dengue virus, but also there's a different strands of Zika virus, right? There's Africa strain, Asian strain, and now Brazilian strain, and the latest is Puerto Rico strain. So we actually have all of them, and the idea is actually compare whether they do the same thing or they do different thing. It's actually very clear the dengue virus do very different effect to the neural stem cells than the Zika virus.
Okay.
So clearly there are differences in that aspect. So one of actually we feel there's another very urgent problem in this emergency is diagnostic. Because currently the FDA or CDC use diagnostic mostly are based on detecting virus RNA directly. And that has a problem because only when the virus are present, detectable in the blood, you can detect them. That has the lifetime of about a week. And that leads to a problem. Many people who actually got infected, they do not know they actually were infected before. So what we're trying working here at Hopkins with a team is try to come up with a more sort of point of care diagnostic tools, which allow us to look at sort of much easier who actually get infected, and also hopefully what develop methodologies allow us to differentiate a dengue virus, for example, from the Zika virus. So that's another clinically very important application need to be achieved.
Of course, that's one that I hadn't thought about. Well, we're reaching the end of our time here, but I don't want to let you go without just asking you to kind of reflect on the last couple of months. What has the experience been like? Working on Zika. You're all doing cutting-edge research in your laboratories all the time, but how has this experience been different?
Well, I might start by saying that it's been really highly collaborative because when we entered the field, we know something about microcephaly, but we know nothing about virology.
Right.
And when it comes to the placenta, of course, I'm even more ignorant, as it turns out. But at least here at UCSF, we managed to get collaborators who are expert in those areas. And so this has become a larger and larger effort, multidisciplinary now. And I think that the rate of progress is really rather breathtaking. It's not something I'm familiar with. Mostly science moves at a much, much slower pace. But right now, this is really turbocharged, and I have to give the journals credit as well, because articles that have to do with this crisis are now being fast-tracked, and they're also being made available right away. That is, they're free access, which I think is really, really important. So I think the scientific community as a whole has really come together in this regard, and I'm sure Hongjun and Guo-li see in Hopkins a similar spirit of cooperation and collaboration that really is wonderful, and I think it's really science at its best.
Yeah, I can completely echo that. So when we started, it's really a collaboration around the U.S., around the states, not just at Hopkins, on 4 different labs. And right now, this number has been increased to 10 labs. We're collaborating with 8 other labs together, try to tackle on different aspects of how Zika might cause neurological disorders.
Yeah, I want to add actually one comment actually was made not by one of the lab members out of our lab. His comment was, normally take our basic scientists years, if not decades, to translate our any findings to a public sort of relevance directly. And this case is actually what's really bringing all the people together. Is basically we can translate whatever we find very quickly, very relevant to public health emergency. I think that's really exciting and keep all this collaboration going.
No, that is exciting. And I think the discussion has underscored the enormous importance of understanding the basic biology that you're working on. On behalf of the Kavli Foundation, I would really like to thank the three of you for sharing your expertise with us today. For our audience, if you would like to watch this webcast again or share it with your friends, it is available at kavlifoundation.org or on our YouTube channel. And to find out about upcoming Kavli Live Hangouts, please follow the Kavli Foundation on Facebook and Twitter. Have a wonderful day, and thanks to the three of you again.
Thank you.